Good afternoon. Good morning and welcome back. I'm John Sullivan, Director of Research at Leerink. I'm pleased to be joined this morning by Edward Lanphier and the team at Sangamo BioSciences. Edward is CEO of the company, and we're joined as well by Liz Wolfe, our Head of Investor Relations at Sangamo BioSciences, and we're very happy that they could join us. This morning, what we'll do is Edward will run through slides for a few minutes to kind of orient you to the company, and then we'll have some fireside chat discussion. We'll be sure to leave some time for your questions before the end of the session. So, Edward, welcome, and why don't you jump into slides?
Thank you, John, and thank you very much for including us in this year's conference. Much appreciated. Did you steal the slide advancer? Oh, I have it. Never mind. So this presentation will contain forward-looking statements. I'd refer you to our Forms 10K and 10Q filed with the SEC. As a matter of fact, we expect to be filing our 10K very, very soon. A couple of high-level comments, and as John said, I've got just 10 slides here to run through, position things. Much more interested in having a discussion around these topics. The most important thing for people who are new to the Sangamo story is our core technology. Everything that we do is derivative of the way in which we're able to engineer a specific class of DNA binding proteins in a way that we can cause them to target exactly the gene sequence we want and drive biologies at the DNA level. That's what's unique about our platform versus any other technology platform. Our goal, and it's the top bullet point here, and it's unbelievably ambitious, is to try and develop this as a new class of drugs. A new class of drugs that functions at the DNA level, and because we're functioning at the DNA level, can actually drive outcomes that you can't drive any other way. And as you'll hear me talk about it, we're actually able, and the goal is not to just develop novel drugs or new improved therapies. The goal really is to engineer genetic cures. And I'll show you why and where we're making that approach. So that's the concept. I'll touch briefly on our business model, what we've done, our capital position, and so on, and intellectual property. But the most important thing for people who are new to the story is our core technology. So this is one quick cartoon just to position it. We're able to engineer, as I said, this naturally occurring class of proteins, zinc finger proteins, so we can cause them to recognize exactly the gene sequence we want, and then we can link that to so-called functional domains that allow us to either turn on or turn off gene expression, or, as I'll talk about mostly, the ability to actually physically change in a permanent way an endogenous DNA sequence. We can either stop that gene from making its normal protein forever, or we can do targeted insertion. We can put whatever gene we want at whatever site in the genome we want. And that really is very important as we start to think about correcting and potentially curing monogenic diseases. So that's the core technology. We've been very successful, as I mentioned up front, in leveraging this, both in the area of human therapeutics, but also from a commercial perspective outside of our core focus, with partnerships with Sigma Aldrich and with Dow AgroSciences in these areas. We also announced about a year ago our first major therapeutic partnership with Shire in the area of hemophilia and in Huntington's, and I'll touch upon those. But really the core for us, the major focus, and I think where shareholders are focused in terms of value creation, is in the therapeutic space. And so if you think about this core competency, and that's represented by the leftmost box here of zinc finger technology strategies, we can apply this directly into a patient, and that's what the top two boxes actually anticipate. We can go into the liver and actually express the normal sequence for factor 9 or factor 8 or lysosomal storage disease. And we just presented data in a mouse model around this at ASH, and I'm, John, happy to come back and talk about that. We can also use this directly into target tissues, such as we're doing in the Huntington's program with Shire. We also have the ability to do this ex vivo, so taking cells out of a patient, modifying those cells, and putting them back. And the most visible of these is our phase 2 programs in HIV, where we're taking out T cells from an HIV patient, putting those back in, and in the goal of creating a functional cure for HIV. So happy to drill down in all of these cases. This gives you a sense of the multiple therapeutic strategies and the approaches, but really all of them have the same central goal. Because we function at the DNA level, the goal is to engineer genetic cures. And so always interesting to see how CEOs try and talk about their company and so on. And so I actually have a new slide for this presentation, John, just for you guys. And it came out a couple of weeks ago and out of the NIH. This is Francis Collins. He's the head of the NIH. And in his blog, he talked about this issue, and I think there's a pointer here, of copy editing the genome, right? Extreme personalized medicine. Ultimately, you might want to replace a disease-causing mutation with a healthy snippet of DNA. You can do this with zinc finger nucleases, and he talks specifically about work that's being done in sickle cell. But this is general. The technology can be designed to any gene sequence. And so Francis goes on to say that this approach of editing DNA is a transformative technology, and that ultimately he expects that we'll develop the ability to edit our own genomes in a safe, responsible way to relieve human suffering and improve human health. So it's not just the Lanphier up here blabbing about engineering genetic cures. This technology platform is being widely recognized, and this is one example, one very, very recent example of a third party, arguably a credible third party, talking about this specific platform and the kinds of outcomes, the kinds of potential genetic cures that can be engineered from it. So the rubber hits the road for us. This is a classic sort of pipeline approach. Let's say it marries the toolboxes that we have and we can bring to bear in the area of HIV, hemophilia with Shire, lysosomal storage diseases behind that, Huntington's with Shire, sickle cell, and so on. In terms of guidance and where we are on these things, we are actively in 2 phase 2 clinical trials with our HIV program. Happy to come back and talk about those. And then we've guided over the next 3 years to 7 new INDs, 3 of which are with Shire, the hemophilia A and B INDs next year, and the Huntington's IND in 2015. But leveraging off this same in vivo protein replacement platform that we're doing with Shire in the area of hemophilia, we can go in and put specifically, or any sequence we want in there. And so we're working on specific LSDs, and again, those were data that were just presented at ASH. And we also have a very active program in the area of sickle cell, which we hope to move into the clinic next year as well. So in terms of near-term catalysts, I think the street's looking for a couple of things from us. First, we'll be presenting data in a couple of weeks at the Conference on Retroviruses and Opportunistic Infections. This will be data from our earlier phase 1 trials and focused on the immunological effects of this treatment, so reconstitution of the immune system in patients upon receiving SB728T. And then the ongoing trials, we've guided to having preliminary data from those in the first half of the year and complete data sets by the end of the calendar year. ASH is a good example of the kinds of meetings you'll see us presenting the preclinical programs at throughout the year. And from a cash perspective, we're in very good shape. Started the year with about $76 million, and we're guiding to ending the year with greater than $55 million in cash. So at a high level, a couple of takeaway points. I think that the key things for us is this is an extremely robust and very differentiated technology platform that has the ability to drive permanent changes in the DNA, which can drive permanent correct protein production. And that allows us to talk about, in the context, as Francis Collins did, engineering genetic cures. We have and have already presented unprecedented antiviral and immunological reconstitution data in the HIV setting. We just presented at ASH a highly disruptive and extremely leverageable approach to expression of any cDNA for protein replacement from the liver. And we have a partnered program in Huntington's. We presented data from that at the Society for Neuroscience last year, and we've guided to having our sickle cell program in the clinic next year. So all of that adds up to completing 2 phase 2 trials by the end of the calendar year in HIV and 7 new INDs by the end of 2015. And the cash guidance that we've given over that period of time is that we expect, after 3 years filing 7 INDs, prosecuting these programs, we expect from where we are now to burn in the range of $30 million. And that's a function of this differential business development strategy and our partnership with Shire. So the last point I'd make, and happy to drill down into this in specifics, is that our goal, and I think we're positioned to create significant near-term value, but the way we're positioned, both in terms of looking at a multiple of therapeutic targets, looking at different approaches, both in vivo and ex vivo, the diversity of our business model, our cash position, also mitigate downside risk. And so our goal is to continue to add value, but at the same time, the realization in drug development that not everything's going to go perfectly. I think we have a balanced effort here. So, John, with that, I'm happy to stop and have a discussion.
That's great, Ed. That's great, Edward. Thank you so much, and thanks for those thanks for those update slides and catching us up on the story in broad strokes. I'll certainly endorse the part of the presentation where you specifically talked about what I think boils down to a pretty shareholder-friendly strategy for in biotech, one where you're able to extend your runway through the fact that it's a platform technology. You didn't talk about your breadth of intellectual property in this presentation, but it's substantive and extends past therapeutics to other fields where you're able to get paid for what you own, and therefore put yourself in a situation where there's less dilution of existing shareholders as you prosecute your therapeutic development plans.
Yeah, and it does come back to the breadth and depth of the intellectual property position, number one. Number two, then, from a technical perspective, the ability to leverage it into any other organism. DNA in us is the same as DNA in plants, is the same as DNA in rats or in CHO cells. And so we were successful in doing a broad partnership with Dow AgroSciences in plants. Not only is Dow using the technology, but they're actually our distributors, they're sub-licensors of the technology in across plants and plant crops. So I mean, literally from flowers to aspen trees to sweet potatoes. They focus on some of the row crops internally. And that to date, I think, has brought in over $40 million to the company. And then it's equally leverageable into research areas and transgenic rats. And we have a wonderful partnership with Sigma Aldrich that, again, has brought in about $45 million to date into the company. And then the partnership with Shire that we announced just about a year ago in the therapeutic space. So it's been it's a diversified business model as a function of the breadth, depth of the intellectual property and the technology platform.
Yep, I agree. And you started the conversation by saying we're trying to do something that's pretty audacious, get the FDA to sign off on a new class of drugs. I would say that it's equally as important to realize that your approach from a business standpoint is one that I think is pretty shareholder friendly.
Yeah.
But nonetheless, the therapeutics programs are very important. Let's start by talking about some of the data that you've seen recently, and maybe you could detail the ASH data
Sure.
a bit for us.
So the concept here, and say it, is in the in vivo protein replacement platform. And if you think about hemophilia, if you think about lysosomal storage diseases, those diseases are treated with recombinant proteins. Biogen makes, Genzyme makes recombinant proteins. Those are provided to the patient. They're infused into the patient, and over time that amount goes down, and then they're reinfused, and it goes down, and they're reinfused. So these are enzyme replacement therapies, protein replacement therapies. In those people who don't have the disease, our cells make a normal level of the protein. So what we presented in ASH was a very general strategy that said we can build zinc finger nucleases that target a gene that makes gobs and gobs of protein, which is a technical term I'll come back to now. And it's the albumin gene. We actually make 9 pounds of albumin a year, and people who are missing one of their albumin genes are perfectly normal. So we make a boatload of this protein. We don't need it. If we just take 1% of the normal production of the albumin locus or gene, we can get more than corrective, more than normal levels of all of those protein replacements. And so what we presented at ASH was taking exactly the same zinc finger nucleases, putting them into a mouse model of hemophilia, showing that we could insert in the albumin gene, the factor IX gene, get normal, 100% normal levels of circulating factor IX in that model, and importantly, from a biological perspective, absolutely normalizing compared to a normal mouse, a wild-type mouse, the coagulation time, so the biological consequences as well. We then took exactly those same zinc finger nucleases that we used to insert the factor IX gene into the albumin locus and put 4 different lysosomal storage disease targets into that and showed exactly the same thing, not the clotting, but that we could express in a very robust way those different gene sequences. So it's an enormously leverageable strategy, in vivo protein replacement platform. And what we've shown is that we can, the goal is to cause a person with that disease to cause their own liver to make normal levels of that protein such that they will not have to take exogenous recombinant protein. And the technology, the albumin gene, once we've made that insertion, is agnostic to the gene we put in, and we've shown that. So it's very leverageable, and if successful, enormously disruptive across a very, very large business right now. So there's been a lot of investor focus on this since the ASH data. It certainly is the piece of work that underpins our Shire collaboration, or the initial part of our Shire collaboration in the area of hemophilia. Shire is funding 100% of all of the work internally and externally at Sangamo through IND filing on factor VIII and factor IX. But as I mentioned, the same zinc finger reagents that we would move forward for hemophilia can be used for any other of these diseases, LSDs and so on, and we own all those. So we can draft on the back of the investment that Shire's making in hemophilia. And so the timing on those is 2 INDs around the hemophilia targets, factor VIII and factor IX in 2014, and then in 2015, 2 LSDs of our own.
Right, and just to be clear, so away from hemophilia is away from the Shire relationship completely?
Yes. Yeah, the Shire collaboration, thank you for clarifying, has initially 4 targets, factors 7, 8, 9, and 10, which are the principal clotting factors in hemophilia. The focus is 8 and 9, but it covers all those. And then they named a 5th target, which is Huntington's disease, which is completely different, one of the other boxes, direct tissue injection into the brain.
Okay, understood. Okay, so away from hemophilia, you have the ability to leverage Shire to prove and then go forward with your own
Yes.
lysosomal storage
We own all of those
candidates. Understood. Okay, let's shift gears just for a minute and just chat about HIV and catch us up on what we should expect out of in development in HIV this year.
Sure. So the nearest term event in that program is the presentation that's coming up at CROI, the Conference on Retroviruses and Opportunistic Infections. I think this is our 3rd year in a row for an oral presentation at CROI, which is a good thing. The focus of this presentation is on phase 1 studies, and in particular, the data showing, and I can say this because this is what we previously presented, data showing that one treatment of these modified T cells, modified in knocking out or disrupting the CCR5 gene in these cells, leads to a long-term durable improvement. The scientists at ICAC use the term reconstitution, but improvement in the overall immune system of these patients. And so in the data that were presented at ICAC, 9 out of 9 patients had an increase in their CD4 counts, which is the target of HIV, and 5 of the 9 actually had a doubling of their CD4 counts.
In the phase 1 studies?
In these phase 1 studies. So the data that will be coming up at CROI is a continuation of that work from an immunological perspective. So
and can you just talk about the safety profile?
Yeah, across all of these, the product has been safe and well tolerated. We haven't had any serious adverse events in this. The most significant are infusion-like reactions, but nothing serious.
Okay, and similar to the Huntington program. I'm sorry, the breadth of the programs with Shire. Are these all in vivo? Is HIV in vivo? Or?
No, this is the lower part of that slide I showed. So this is the ex vivo modification of autologous T cells, so CD4 cells from an HIV-infected patient. Bring those in the laboratory, knock out the CCR5 gene, reinfuse those cells back into the same patient, the goal being to show both an antiviral effect as well as an immune reconstitution event. So the data coming up at CROI will focus on this immune reconstitution aspect of the disease. Data coming later in the year, preliminary data in the first half of this year, and then complete data sets from by the end of the year, we'll focus on our ongoing studies, which include a treatment interruption during that period, so off of heart, and that's where we'll be able to evaluate the antiviral effects of the drug.
Understood. Okay. Let me just do this as we get nearer to the end. Questions from our audience before we continue? We have a microphone in the back. Edward, maybe you could just talk a little bit about, let's just circle back to the intellectual property and kind of the protection that shareholders get from the intellectual property estate of the company.
Sure. It goes, I'll start with the historic because that really built the foundation. And
if you don't mind, I'm asking also because as you've been at this doing good work for several years, investors may wonder how close are we to getting to the end of the intellectual property estate?
Yep, and a great question. So it starts with the original academic IP, which was filed in the mid-90s. We went out and exclusively in-licensed that from the main groups that were doing that, all the groups that were doing that. And subsequent to that, we've been filing multiple new patent applications. And those relate to both the design and composition of matter of engineered zinc fingers, the type of linkers that you use, the type of delivery you use, the gene targets that you go after, the therapeutic outcomes. So it's really belt and suspenders. And as we move forward with these, we continue to file, especially around specific targets. So the targets relating to therapeutics are really going to continue for quite a while out there. But I think the best evidence, if you will, of the strength and breadth of the IP is that the only people who are able, companies that are able to do anything commercially with anything in the zinc finger space are ourselves, Sigma, and Dow or Sigma's licensees, or our licensees like Shire in the space.
Okay. Can you just, any questions in the audience? Maybe you could just talk for a second, as this is a new class of drugs, and there's probably a significant effort involved in getting clinical trial sites, and it would seem to me that there's a lot of persuasion that has to go on. Maybe you could talk about, as you contemplate your clinical development plan, how is it that you advance these candidates, and how is it that you sign up the sites that you need and that sort of thing?
Yeah, I mean, I'll use the HIV program specifically, and I can talk about going forward, but we've been inundated with interest. I mean, the goal around the HIV program, and from a patient perspective, from an advocacy perspective, from a clinical perspective, is really the next step in HIV. It's not a novel therapy. The goal is a functional cure. The goal is to reconstitute a sufficient number of cells in patients that cannot be infected by the HIV virus, but can mount an antiviral response to the virus, with the goal being a sufficient amount of time, a sufficient amount of numbers of these cells to cause these patients to become aviremic while not on heart, so a so-called functional cure. And that is of enormous interest. So we've had zero problems with A, clinical sites being interested in this, and B, patient interest in the trials. As we think about moving forward into the protein replacement areas, there is we all know about the costs of these drugs, hundreds of thousands of dollars a year, and we already know from reviewing this with in market research with third-party payers that if we can develop strategies that are potentially curative in a permanent way and reduce that lifetime of hundreds of thousands of dollars, which ultimately ends up with millions and millions of dollars in an individual patient with a single treatment up front, there is enormous interest in both from a regulatory perspective and a payer perspective. And as payers become a bigger and bigger component of the drive to get drugs not only approved but reimbursed, there's an awful lot of interest in this as well.
Okay. And as investors think about the steps toward development in protein replacement,
yep.
Is albumin the key inflection point, or do you need analogs to albumin in different enzyme replacement programs? How should they think about that as a point of progress?
Yep. So the albumin site really is, and all the data suggests that we have now, that albumin is completely leverageable, that it is absolutely 100% agnostic to whether it's the factor 9 gene, the factor 8 gene. Gauchers, Pompeys, Fabres, Hunters, Hurlers, and the list goes on. The point I'm making is that the insertion with these, quote, homology arms that we put around it is agnostic to the gene. So our goal, once we establish this in the clinic, is to leverage the hell out of that going forward. And there's enormous leverage not only from a scientific perspective, but from a regulatory and manufacturing perspective. It's the same zinc finger nucleases that we've putatively, in this case, gotten established for factor 9 or factor 8. Well, we've already done the master cell bank. We've already done the GMP manufacturing. We've already done the toxicology studies for that. And so it's highly leverageable going forward from both a technical but also from a development perspective.
Great. Last chance for a question. Okay, Edward, go right ahead, sir. Could you repeat the question?
Yeah, the question is, if there's a functional cure in HIV, will these patients still be transmissible? So it's a great question. There's a distinction between a, quote, functional cure and a sterilizing cure. Sterilizing taking the point where you cannot find any existence of the virus in any part of the patient. A functional cure says that the immune system is controlling the virus in a way that the immune system is healthy and reconstituted, and that the levels of HIV are below detection such as they are on heart. That doesn't mean that every single cell in the body, the virus has been eliminated, just as it's not with heart.
I'm afraid we're going to have to leave it there. Edward, congrats on Sangamo's progress, and look forward to hearing more soon.
Thanks, John.
Thank you.